Good morning, everyone. Welcome to day 4 of the conference sessions. So now I'd like to invite, she's already here, but thank you, Dr. Goyal, for chairing today's session. We'll start.
It's my privilege and honor to start today's session by introducing Dr. Maria Tenje. Maria is the group leader of the EMBLA, the Enabling Microtechnologies for Biomedical and Life Sciences Applications Group at Angstrom Laboratory at Uppsala University. She received her PhD from the Technical University of Denmark. Her research group focuses on developing miniaturized systems for biomedical and life science applications based on sophisticated micro and nanofabrication methods and the use of microfluidics. They focus on organ-on-chip and droplet-based microfluidics in projects funded by the Knut and Alice Wallenberg Foundation, the European Research Council, and VR. Her talk today will focus on microfabrication of 3D cell culture scaffolds. Maria, it's a pleasure to have you, and I'm greatly looking forward to your talk.
Thank you very much, and thank you for that introduction, and thank you for inviting me to speak here today. Let's see. I hope that you can see the slides in the way that you are supposed to see them. And let me just also get a laser pointer. Yes, so thank you very much for having me here to speak on my favorite topic, which is microfabrication, and especially then microfabrication of cell culture scaffolds. But just like it was said in the introduction, I have a background in microfabrication. I actually did my PhD in fabrication of mechanical sensors. But I have then moved my research focus from the more purely focused on engineering and technical aspects towards life science applications. But still, my interest and my motivation is really to provide sort of engineering and technical solutions to the different questions to address. Having said that, I shall get started on my talk. And the outline is basically to motivate and to why we want to do this type of research. I should say also, yes, I am based at Uppsala University, but this year I'm actually doing a sabbatical at the University of Santa Barbara in California, also to learn some new fabrication techniques here. But I'm going to focus a lot on my presentation on classical microfabrication methods and to explain how these can be used in the organs-on-chip research area, either for 2D chemical functionalizations or modifications, or to do 3D engineering of scaffolds, of cultured scaffolds. And then, of course, wrap up in some conclusions and quickly address some remaining challenges. And I have really tried to save enough time for questions afterwards, and maybe there we can go into more details on the different challenges of the field. But I would like to start by simply asking the questions or addressing the aspect, the importance of the 3 dimensions. Because we know that in cell cultures, we normally use them in the 2D format, whereas cells are sitting on a flat surface. And this one is true both for classical cell cultures using petri dishes and culture wells, but also in many of the organs-on-chip systems developed, the cells are still stuck on a 2D surface. And importantly here, for example, the stiffness of the surfaces that the cells grow on is usually very high, gigapascal. And it means that there's a forced apical basal polarity. The cells can only adhere on one side of the environment that they are in. So they are restricted to this XY plane. Usually they are not constrained, so they can move freely across this area, but only in the 2 dimensions. And then if we look at the situation in the body, obviously cells are then actually growing in 3 dimensions. So first of all, we don't have this no polarity, no pre-described polarity for the cells. They are now able to go in all directions, although they are more limited on how much they can migrate in this scaffold, because they are hindered both by sort of components of the scaffold and the fact that they need to remodel and interact with it. But more importantly, the stiffness of the environments that the cells normally encounter in the body is on the lower stiffness, so say kilopascal, compared to the gigapascals that you have on the harder substrates. So if we want to now go from the 2D to the 3D, that means that we need to develop methods to be able to control the physical properties. And with this, I mean both the mechanical properties and really the dimensions of these scaffolds. And we want to also be able to control the chemical properties so that we can design scaffolds in a suitable way. And we want to design them most often so that they mimic the body. And the first question that we need to ask ourselves is then, what material do I want to work with? Because that determines how I can, what mechanisms can I use for cross-linking to make sure that some parts of this material will be rigid or stable over time. And do I want to support cell adhesion, or do I actually want to inhibit cell adhesion? Do I want to control where the cells can be in this matrix and how they can migrate? Second question you need to think about is, what dimensions am I interested in? What do I want to study? Do I want to study behavior of single cells or interactions of maybe two cells in a very direct communication? Or do I rather want to study for a population of cells? And do I want to perform some assays later on off-chip that requires maybe a large number of cells to have sufficient amount of sample for my analysis, if I want to do some RNA sequencing, for example? So these are important questions to ask early on. But usually, we will end up working with a material that is a hydrogel. And a hydrogel, that is a cross-linked hydrophilic polymer chains that within their network can hold large amounts of water. 95% of these scaffolds consists of water. And because they have so much water in the scaffolds, the properties are very similar to the tissue that we have in vivo. And on the image here, you just see jello, formed into a sort of dessert. And I think this is a material that you can relate to. So a very soft and jelly construct. And that's a typical hydrogel. And hydrogels are important because, as I was saying initially, we need to think about the mechanical properties and the stiffness of these materials. And the classical materials, polystyrene, culture plates, or glass slides that we could grow cells on, they have elastic modulus and properties that are very similar to that of bone. But if we want to mimic like the brain or a lung model, for example, then we need to work with softer materials down on the kilopascal range. And that's where this gummy bear, the hydrogels, fit in very well. So at least we can agree that we want to work with a hydrogel, but it's a large group of different materials, and we need to specify a bit more clearly what type we're interested in. We can classify hydrogels into two different categories. So the ones that are naturally derived, that they are simply derived from tissue. And Matrigel, it's a material that probably most people who do cell cultures have encountered and have used. We have agarose, that's also commonly used, collagen or the hydrolyzed form, gelatin. We can have fibrin gels, chitosan silk, hyaluronic acid. These are all types of naturally derived hydrogels that are often used for cell cultures. And the advantage of the naturally derived gels is that they already have adhesion motifs that the cells can adhere to, that they can interact with. cells, they grow very well in these gels. But the challenge is that we have large, I mean, first of all, you need an animal to get access to the material. And that's, of course, problematic if you are working towards replacing animal experiments and the use of animal in research. But it also means that you have large batch-to-batch variations, and you can't actually know exactly the composition. Matrigel is, for example, a mixture of many different types of materials, and it varies on batch. So in that respect, it's interesting to turn to synthetic hydrogels, so polyethylene glycol, for example, or polyacrylamide, that you know exactly the structure of, and you can then modify this backbone with addition ligands, for example, for the cells to adhere to, or other sort of active components that you want to introduce into your matrix. So you have much more control if you work with synthetic hydrogels, but you have also none of the naturally occurring chemical groups that the cells would like to see. So you need to modify your gels to support cell addition. But all of these gels, so irrespective if you work with a naturally derived or a synthetic gel, you need to cross-link it so that it becomes stable. And there are different ways to do that cross-linking. And I would like to show you 3 commonly used different cross-linking strategies. So the first one is simply to use thermally cross-linking, and that you can use for matrigel and for collagen, for example, because you have these polymer chains, and if you change the temperature, cool down, for example, they will come together and they will form a network. So that's in a very simple setup. That's a very convenient way of achieving the cross-linking. It happens rapidly, you just change the temperature. On the other hand, you don't have any spatial control. The whole material will cross-link. And also, if you have encapsulated cells and you need to go to very low or very high temperatures, you may actually affect also those cells. And it also means that the cross-linking it's not a covalent bond, so it easily degrades. And if you have small temperature variations, that can then also change the cross-linking density of your gel and hence change also the mechanical properties. So although attractive, it has some sort of severe disadvantages. You can work with ionically cross-linked polymers, and here the classical example is alginate that is cross-linked by the introduction of calcium ions. You don't need to perform any chemical modifications of the polymer, so it's also very easy to use. But the way it works is that you have your hydrogel and then you have a solution where you have your calcium ions. And the ions will diffuse into the network. And as it diffuses in and meets the chains, it will form the cross-linking. And that means that you will actually have an inhomogeneous cross-linking density. The outer regions will be more cross-linked than the core. And if you make a very thick slab, the core might not be cross-linked at all because you have the diffusion limits. And likewise, these gels can be quite easily degradable. If you lose the calcium ions, it will break apart immediately. So to form very stable cross-linked gels, covalent bonds are attractive, and to use a radical cross-linking mechanism is the most common way, I would say, to use hydrogels together with microfabrication techniques. And here we can use, for example, PEG, a synthetic one, or we can use gelatin, a naturally derived one. What is required is that you incorporate functional groups on the polymer backbone that can be initiated to form a cross-linking network. And acrylate groups is, for example, very commonly used. What's shown here in the schematic is that you have your photo initiator or your cross-linking initiator that gets initiated by an external stimulant. Here, UV light it's conventionally used. And that then activates your initiator, forms radicals that start interacting with your polymer chains, and then actually that forms a chain reactions. So it interacts, it forms bonds, and it moves on to the further polymer chains. If you use UV light, you can achieve a very high spatial resolution. It's a fast reaction, and it provides a long-term stability to your polymer, cross-linked polymer network. There are some advantages, and cytotoxicity is one of them. Either the radicals used for the cross-linking can be cytotoxic to some cells that are encapsulated already in the matrix. And also the initiator, the UV light that you need to first expose the gels to, may also have negative effects, depending on which gels you have encapsulated in the scaffold. Also, the bonds that you form, since they are covalent, they're very stable. So if you cross-link too much, that may prevent the cells to remodel and sort of move around and interact with the matrix. But that's, of course, something that can be tuned during the fabrication. So we have now looked at different types of materials that you can use and how they can be cross-linked. And now I want to show you actually the techniques to use. And we are moving into the clean room. Because for microfabrication, you normally work inside a clean room to make sure that the fine structures you want to define are not disturbed by dust or other particles, larger particles. And I'm going to show you some of the very classical techniques that we used and show how that can be worked for this type of application. But you know, the different techniques that were developed for microfabrication in the 50s and the 60s, here the motivation was to make miniaturized electronic circuits that now has enabled our, portable computers and mobile phones and everything. And on the two SEM images that I have here on the slide, you can see human hair, so about 100 micrometers in width, on top of a 4 megabyte memory chip. And you can see these small trenches that have been defined with the microfabrication techniques. On the other picture, there's some red blood cells, 5 micrometers across, for example. And you can see that the features that we can define with microfabrication is really sub-micrometer. But what you need to remember that even if the fabrication techniques that we use has resolution on sub-micrometer scale, hydrogels can normally not be fabricated to that degree of resolution because they will swell and they will interact because they have all this water inside the polymer matrix. It will change a lot in the dimensions. So normally we are operating on them a couple of tens of micrometers, maybe 50 micrometers and upwards. And that also usually makes sense because if you want to make an organism-on-chip device, for example, you want to culture many cells in there. And you don't want a scaffold that is on the subcellular range of dimensions. But it's just important to remember that it's not the techniques itself that sets the limits of what resolution you can obtain, but it's rather the material that we are working with. And for microfabrication, really the workhorse is UV lithography. And just very briefly, imagine a cross-section, you have a substrate, you put a layer on top that is sensitive to UV light. You can bring in a mask and you expose to the UV light, and that can either form bonds if you work with a negative resist or a negative hydrogel, or it can break bonds if you work with what we call a positive resist, or then a hydrogel that has been functionalized so that bonds cleave upon exposure to UV light. And that's which approach you use. It's rather a matter of preference and how you want to modify your gels. But you can then use this technique either to vary the topology, to actually form sort of different heights. And on the images here, we have just fabricated some simple squares, and we could see the cells, and we could see that the cells were sitting on top of these structures. Or you can use the method to functionalize, chemically functionalize certain areas so that the cells will adhere there, not in others. And these are some images from work in my lab, and I want to describe now a bit more on the work that we have done. I quickly just want to show you the different equipments that we can use for this. And you can work, if you go to the clean room and you work with UV lithography, you will need a mask aligner, which is the type of equipment where you can insert your mask, you're exposed to the UV light, and you get your defined structures. You can also work with maskless aligners, where the technique is rather laser-based. And then we have spatial light modulator that determines where the light will be exposed onto your substrate, and that forms the pattern. In the work that we've done, we simply just used a sort of a homemade setup with the UV LED, a 3D printed holder, a mask, a glass slide with some metal pattern on, and below the substrate. So it doesn't need to be more complicated than this. But there are also commercially available systems that you can dock onto a microscope and thereby control the exposure on your slide that you place on your microscope. The two first equipments, that's for sort of large scale, for fabricating a lot of patterns. But if you are just interested in research questions, the homemade system works perfectly well. So in the work that we have done, where we use the UV lithography to pattern a hydrogel, I want to describe the work on selectively functionalized hyaluronic acid. And hyaluronic acid is a naturally derived hydrogel, does not support cell adhesion. So if you want to functionalize it for cell adhesion, you need to, that's one of the features that you can control. Hyaluronic acid is found in various tissues in our bodies, the joints and the brain, and that's why we have chosen to work with this material. We started by functionalizing with acrylamide groups onto the backbone, and then we could place a small amount of hydrogel in a mold, press down a glass lid so that we define the height, expose it to UV light, and then have a cross-linked hydrogel. On top of this cross-linked hydrogel, we then placed RGD solution, which is a peptide to support cell adhesion. And again, we exposed to UV light, but this time through a photomask, where some areas are open to the light and some are blocked. And in that way, we could generate a pattern of functionalized hydrogel. So the RGD then bound to these acrylamide groups in the areas exposed to UV light. By increasing the exposure dosage, we bind more RGD to the scaffold. And we could make structures down to 5 by 5 micrometers, going up, say, 200. You know, there is basically no limit to how large structures you can. To have cells to adhere, we need to have a minimum structure of 25 micrometers. And on the larger pattern areas, more cells would adhere. And again, increasing the exposure dosage increases the amount of RGD that we can bind. And increasing the exposure dosage, we could also increase the stiffness of the material. And we used this platform to study how brain endothelial cells behave when they are cultured on this hydrogel, but their adhesion is constrained. So we started by forming lines of different widths, going from 10 micrometer up to 100 micrometer. And that's what you see here, the green lines of the RGD patterns. Then we seeded brain endothelial cells onto these lines. And here we have stained them for F-actin and the nucleus. So looking at the actin filaments. And we also stained for ZO2, ZO1. So a tight junction protein that that sits on the cell membrane, so that we could identify the cell body shape. And looking at specifically at and comparing the thinner lines to the broader lines, you can see quite a few differences between these. So first of all, there's obviously fewer cells on the thinner lines, but also the cells, they sort of align, so they fit just one by one next to each other, and they form a very thin structure. Whereas on the thicker lines, you have multiple cells that sit next to each other. So looking at these two extremes, we wanted to investigate the cell body and the nuclear orientation and the cell body and nuclear elongation. So alignment is simply a measure of, does the cell body realize that there is a line, and how does the line control the alignment of the cell? And if we then looked at the cell body for the different line widths, with the non-patterned control surface where there is RGD everyone and the cells are not constrained to move, there is also no preference in the direction. And here we're looking plus minus 90 degrees. The cells are just going in all directions. But as the line width gets more and more narrow, the cells are approaching zero alignment, so being fully aligned in the direction of the RGD pattern, and the spread also decreases drastically. And the same goes if we look at the cell nuclei. And it's not surprising because the cells are not able to interact with the matrix outside. They need to just stick to this line. If we then look at elongation, and here this is a measure of the eccentricity, so a line is 1, a perfect circle is 0. We wanted to see, okay, so how does the cells, sort of what happens to the cell structure? And again, looking at the cell body, we can see that as it goes from the non-pattern to the more thin lines, it goes and it approaches 1. So it gets more and more flat, i.e. more and more elongated. But it actually becomes more pronounced when we look at the shape of the nucleus, which is much lower in the non-patterned control sum. And then it approaches 1 or gets more and more flattened out as the line gets thinner. So the cells are very clearly affected by the fact that they are sitting on a line and they are moving and adhering to this structure. What we also then started to realize looking at these images is that if you see, for example, this nucleus here, it's tilting a bit upwards, whereas, for example, this nucleus, it's actually tilting a bit downwards in the direction of this image. So we can then determine the orientation of the cell body and the nucleus to be either negative, downwards, or positive, upwards. And when we went through the data and we looked at all of these, we could see that there was a clear preference for a negative orientation, both of the cell body and the nucleus. And this is something that is referred to as chirality. There is an orientation preference of these cells. And that's an aspect that's very important in developmental biology, because we are not symmetrical as we develop. The cells need to get cues that breaks the symmetry so that the organs can develop properly. And we could see that these brain endothelial cells also had a preference in their orientation. You can see that if you have a non-patterned surface, there isn't a preference. And as the line, it's mostly pronounced in the 100 micrometer wide lines, and it gets less and less pronounced at the cells as the lines either get thinner or as they get wider. And that's because you need to balance two aspects. So first of all, the cells need to know that there is a border. They need to know that there is a directional cue. On the non-pattern, there aren't any cues, so they have nothing to align themselves to. At the same time, they also need to have space to be able to move. And on the thinner lines, there isn't any space because they are just constricted to this structure. So that's why we see the largest expression of the chirality on the 100 micrometer wide lines. And we could even zoom in and sort of split this 100 micrometer wide lines into 5 different regions. And remembering that they have a preference for negative orientation, we could see that preference was expressed mostly in the center region. And that's exactly what I was saying. They need to have space to move. And even looking at the top and the bottom region, so region 1 and 5, it's interesting to see that in the fifth one, they actually have less degree of expression. And that's because they are sitting at the border. They understand that there is a line. That's a cue to orient themselves. They want to go negative, but they can't go outside the line. In the top region, however, they get the cue and they are able to move because they have also RGT in that direction. So it was very interesting to see how the cells are so sensitive to the mechanical cues that they get from being physically confined on this matrix.
Maria, just to remind you, of 5 minutes.
Yes, I just realized that. So thank you also for the reminder. And actually what I would like to do in the interest of time, because as I was saying, when you're exposed to UV light, you also affect the mechanical properties. So there is, as an alternative to functionalize chemically, one can work with a technique that is called microcontact printing, where you simply transfer the chemicals without using UV exposure. And I wanted to discuss this one in a bit more detail, but I'm going to skip that actually, because I would like to talk about 3D structuring and just give you one example of a new interesting technique for that. Because I think when you think of 3D structuring of hydrogels, you think about bioprinting, for example. And you can use many different types of bioprinting, inkjet-based or orifice-free based or extrusion bioprinting. But I don't want to talk about the bioprinting because that would be a full lecture in itself. But you can use molding, and that's an example that I would like to describe to you. Because just like you can use soft lithography to prepare the stamps for the chemical patterns that you use in microcontact printing, you can also mold hydrogels. So you simply form the inverse of the structure that you want to work with. Here it's been defined in silicon. And then you can pour your hydrogels or agarose, including cells, on top of this structure, and then use the temperature or the calcium ion approach or UV exposure to cross-link the structures. And we have used collagen molding to form very thin sheets. So we can dissolve the collagen first in acidic conditions, but then bringing it up to neutral pH, we can introduce cells into the suspension and then gellify it at 37 degrees. And during the gellification process, we can actually take out some of the water and compress this structure into a very thin sheet. And with that technique, we have formed collagen sheets on the width of 50 micrometers only, with cells inside. And we could introduce this into microfluidic chambers and culture them here. But just to show you also that collagen can also be used for much larger structures. And I think a very elegant work has been done by Stephanie Decroix and her research group. And I know that she has already given a lecture in the course, so she probably talked a lot about the details of how they have molded collagen replicas of the villi from the gut. And I think this is very cool technique where they use a bronze mold. So here, rather forming structures on several hundred micrometers in dimensions. But again, just having a mold, putting the collagen with the encapsulated fibroblasts inside, and then forming the structures. So the molding is a very effective technique, both for thin structures or for thick structures. But my favorite fabrication technique for 3D microfabricated scaffolds is really two-photon polymerization. That's quite a novel technique, and it enables free-form fabrication possibility because it's laser-based, and you can then expose a material and sort of initiate this cross-linking. You still require the photoactivated initiator to be mixed inside, but the advantage is that if you compare to UV exposure, then you have a column of light that goes through the whole part of your matrix. Whereas if you work with two-photon polymerization, it's only at the voxel, at the focal plane of your laser, that you will initiate cross-linking. And that means that you can write structures inside the gels with a very high resolution and accuracy. And just like you can do UV light exposure either in negative or positive approach to either form structures or remove structures, you can use two-photon polymerization to cross-link hydrogels. Here they're written as scaffold, and then the encapsulated cells in these scaffolds that could grow. Or you can use the technique in ablation. So simply have a hydrogel with cells inside, come in with your laser and write the structures that you want to have.
1 minute.
For example, a vessel structure that can then be where the cells can grow, just on the dimensions of a microvasculature. Which brings me to the summary of the lecture, where I want to say that microfabrication techniques that were originally developed for MEMS and electronic circuits can be used to control the chemical and the physical properties of hydrogels, and that's very attractive to make well-defined organs on chip model. And the main limits of this is really the swelling of the hydrogels. That's what limits the resolution, not the fabrication techniques. And of course, you need to think carefully about what material you want to work with, but you can structure both in 2D and 3D. And I would say that this long-term stability and the biocompatibility of the chemistries, the functionalization chemistries, these are really the current main challenges. And as always, there's so much more that I want to talk about than what time allows for. But if you're interested in further reading on this topic, there's a review article from my group that was published a couple of years ago in the journal Organs on a Chip. It's published open access, so you should be able to find it. If you can't get hold of it, please just get in touch with me. I'm more than happy to send you a copy. And with that, I really say thank you to my research group who is working on this with me, to our funding agencies, and many thanks to you for your attention. And if there is some time left, I'm happy to answer any questions that you may have. Thank you very much.
Thank you for that wonderful lecture. We have time for questions, so yeah.
Thanks a lot to Dr. Maria. It's a very interesting talk and a useful talk. I have a small question. As you described that we can use natural hydrogel as well as synthetic hydrogel. So based upon different types of cell we use, can we mix them and try if it works for us? I just need some suggestions from you. What is your opinion?
Yeah, so to combine natural hydrogels with synthetic hydrogels.
Yeah. Will it work in better
that
way?
That is definitely possible to do. And for example, in some of the works that we have done, we have used PEG as the main constituent because we can control the structure very carefully, but PEG is not cell adhesive. So we have simply mixed in some gelatin to provide an environment for the cells that they can adhere to and sort of interact with. in the work that we did here, we simply mixed the gelatin, so it was not bound to the PEG scaffold, but it was just physically constrained within the polymer net. So in regards then to stability, the gelatin eventually leaks out. So there is a benefit if you can really link it to the scaffold, but definitely you can mix also naturally derived and synthetic gels, yes.
Thank you so much.
Hi, Maria, nice talk. I just had a quick question. In one of your slides, you were showing about the degree of elongation on different contexts. I mean, the size of the channels itself, 5 micron, all the way varying to 100 and non-preferential one. So I saw that in the 5 micron, of course, it was tending towards 1. But as you move across the spectrum from 50 micron to over 100 and non-1, it still goes back again to 1. Like it dips in between and then goes back to. I was just wondering what's the reason about behind that.
Yeah, and that was very well spotted. And I would say that's actually something that we're still trying to understand. Why does it act? And you could also see that there is a difference of how much the cell body elongates and how much the nucleus elongates.
Yeah.
And we are also trying to understand what is, sort of what moves first. Because one would expect that it's the cell body that first senses the lines, and then the actin would sort of push down on the nucleus and orient the nucleus. But when we are digging deeper into the data that we have, it doesn't actually seem to be happening that way. So we are still very puzzled about how the cells realize that they are on a constrained surface.
I see. Very interesting.
So yeah. So there is more to learn about on this topic.
Thank you, Maria. Very nice presentation. I have a question related to the selective functionalization. Is there a role with the degree of functionalization? Because I did not get from your talk, how much are you functionalizing the alginate? Is it like 80%, 50%? 100%?
Yeah. So the work that we've done on hyaluronic acid. So first we need to functionalize the polymer backbone. So it has these active groups, so that we can form the cross-link. Is it this functionalization you had in mind, or is it the binding of the peptide?
No, it's that functionalization, because what I believe is you may not be having it on the backbone, but on the side chains, right? Because you can only have acrylamide groups coming to your side chains rather than to your main backbone. Am I correct?
You could probably be correct. I have been told it's sitting on the backbone, but I'm not a trained chemist. So if you correct me on this, I'm more than happy to expect.
I think so, because it cannot be on the backbone. It has to be, because I do not know what kind of acrylamide is used, but it has to be something to do with the side chains, so that they are hanging on to respond to the UV.
Yeah, but it's metacrillate groups that we are adhering, and I'm referring to the backbone of the hyaluronic acid structure. And the degree of functionalization, I think it's about 30% that we have.
Okay, and have you tried to see if it varies? How does the whole thing shift? For example, if you go beyond 30% or lower than 30%?
Yeah. So both the degree of functionalization of your polymer backbone, but then also how much photoinitiator you mix into your solution, as well as how much UV light you expose the matrix to. All of these three factors determines how much cross-linking you will achieve. And actually adding more photoinitiator does not necessarily lead to a stiffer gel, because if you have more photoinitiator, you just get a more rapid cross-linking.
Yes, this I can imagine, because you have to finally cross-link your 30% that is onto your backbone. So it has nothing to do with the amount of initiator that you're adding, because probably the initiator is decided onto how many cross-linkable groups you want to cross-link. And some probably you want to leave them hanging on. They don't bother you anyways, right? For For example, photoinitiator, if it's 10% of cross-linking, the rest 20% of your acrylamide groups are just hanging on there, probably because they are also biocompatible.
In the interest of
Exactly.
giving other folks a chance to
Okay.
ask some questions,
Sorry.
I
Okay,
will
thank you very
request you
much,
to
Maria.
continue this discussion on email. I believe there are time for the two last questions here. Yeah.
Hi, Dr. Maria. Thank you for the talk. So my question is relating to the peptides, specifically RGD that you had shown. So I guess this is a good approach to mimic other extracellular components using the binding domain. But how generalist is this approach? Can you use these sort of short peptide fragments for other extracellular components as adhesion points for cells? And if you do this for more other extracellular components, do you lose out on other long range properties like viscoelasticity and other things? And what is the effect of using this?
Yeah, no, so definitely, I mean, using the peptide is not the same as having the full proteins. But what we have looked at is also RGDs, simply because, it's very commonly used and many cells have ligands to adhere to it. So it's recognized cells. And in that sense, it's very attractive. We have also worked with functionalizing with laminins, for example. But since those proteins are so sensitive, it's more difficult to actually make it work and to have the functionalization sites still active. But we looked also at RGD and sort of linking it to these acrylamide groups with either a short linker or a longer linker. And we could also see that cells were preferring or binding more strongly if the RGDs was using the longer linkers. So there is something with steric hindrance and how the cells interact and sort of the space that the cells themselves occupy. But RGD functionalization of different materials, it's been done with several other groups also, and it's a quite convenient way to control cell adhesion.
Do you know of any other kinds of components that people have tried to mimic with just peptides, or do people generally go for the full-length protein like collagen or any
No,
other?
so for the laminins, for example, there's a sequence, this ICBAB, that's a peptide sequence that you can use to model laminins, for example. And there's many other peptides.
Okay, thank you.
Hello. Yeah, thank you, Dr. Maria. It was a wonderful talk. So, my question is that if we mix 2 or 3 components like hydrogel or collagen, and then we cross-link them in the device, because we have been facing some issue that it is not sufficiently creating a hindrance or a blocking for us if we are using even a combination of such gels. So, if we cross-link them, will that help, or would it provide a better stiffness for the gels which we are using if we mix and cross-link those material?
Yeah, so if you cross-link them, you will form a more stable gel, and you can. So, do you want to hinder movement of cells across in this, or?
Yes. So in the channels, essentially our hydrogel will be filled with cells, and I don't want that to cross in my next compartment. Whereas, so with that we are facing a lot of issues. So that is why if we cross-link those gels, will it help, or in what sort that can provide a better stiffness?
Yes, so then you will constrain the cells more into that compartment. What you need to be careful with is that you don't constrain the cells too much, because 3D cultures of cells is very much, the cells need to be able to model and remodel the matrix and to move around in the matrix. And it's very easy. If you have cells and you capsulate and you cross-link the gel, you see the cells are still alive, they're happy there for a couple of days, but then you just have them as round lumps and they die. So there's a fine balance for you to find.
Thank you, Maria. That was a wonderful presentation and a wonderful discussion. Thanks so much for joining us. And we would have loved to see you in person. Thank you.
Yes, next time. Thank you very much. Thanks. And don't hesitate to get in touch if you have other questions. Thank you. Bye-bye.
We'll break for coffee now.